the skeletons tell a different story than the one we have been led to believe. For centuries, historians pointed to Asia as the birthplace of the greatest pandemic in human history, the Black Death. But deep beneath the soil of a remote village in Central Asia, ancient DNA has been patiently waiting, harboring secrets that would rewrite our understanding of the deadliest chapter in human history. Imagine standing in a 14th century village as a strange illness begins claiming lives. First one death, then 10, then hundreds. No one knows what causes it. No one knows how to stop it. The dying gasp for breath as black swellings appear on their bodies. Within days, half the village lies dead. Within years, half of Europe. What unleashed this catastrophe that killed as many as 200 million people? Where did it truly begin? And could it happen again? For almost 7 centuries, these questions have haunted us. Now, in a groundbreaking fusion of archaeology, history, and cutting-edge genetic technology, scientists have uncovered evidence that challenges everything we thought we knew about the Black Death's origins. The answers were buried in the teeth of the dead, preserved in microscopic fragments of bacterial DNA that survived for nearly 700 years. This is not just a story about plague. This is a story about scientific detection, about following a deadly trail through time, about how tiny strands of genetic code can solve one of history's greatest mysteries. It is about finding patient zero of the deadliest pandemic in human history. The truth, as it turns out, is far more surprising than the myths we have been told. The year was 1346. Merchants traveled along the Silk Road, carrying luxurious fabrics, exotic spices, and unknowingly, something far more sinister. A disease that would soon bring the medieval world to its knees. By 1347, Italian merchant ships returning from the Black Sea docked in Sicily with dead and dying men aboard. Their bodies bore strange black swellings the size of eggs in the armpits, neck, and groin. The swellings oozed blood and pus. Fever, chills, and excruciating pain followed. Death came within days. This was the beginning of the Black Death, a pandemic that would sweep across Europe, the Middle East, and North Africa, killing between 30 and 60% of the European population alone. It remains the deadliest recorded epidemic in human history. For centuries, we have accepted a particular narrative about where the Black Death came from, explains Dr. Isabella Blackwell, a paleogeneticist at the University of Cambridge. The traditional story points to China or Central Asia, with the disease traveling westward along trade routes until it reached Europe. But science has a way of challenging our most firmly held beliefs. Dr. Blackwell stands in her laboratory surrounded by advanced DNA sequencing equipment. On her desk sits a small plastic container holding what appears to be a yellowed tooth. This, she says, lifting the container, is how we rewrite history. For most of recorded history, the origins of the Black Death remained a matter of educated guesswork. The earliest accounts place the outbreak in China or somewhere in Central Asia in the 1330s. Historical records speak of a great pestilence that killed millions across Asia before reaching Europe. The standard narrative suggests that infected rats and their fleas traveled aboard merchant ships and caravans, spreading the disease across continents. But there was a problem with this story, a lack of hard evidence. Historical accounts can be unreliable, explains Professor Thomas Kaplan, a medical historian at Oxford University. Medieval chronicles often mixed fact with fiction, observation with superstition. And for many regions in Asia during this period, written records are scarce or non-existent. Without physical evidence, we could not know for certain where the Black Death truly began. This evidentiary gap persisted for centuries. Then, in 2011, everything changed. A team of archaeologists excavating a series of burial sites near Lake Issyk in what is now Kyrgyzstan made a remarkable discovery. They uncovered a medieval cemetery where an unusually high number of burials dated to 1338 to 1339, nearly a decade before the pandemic reached Europe. Even more intriguing, the headstones carried inscriptions explicitly stating that the deceased had died of pestilence, an apparent epidemic that struck years before the European outbreak. Could this be evidence of the Black Death's true point of origin? To answer this question required a marriage of modern technology and ancient remains. Enter the field of paleogenomics, the study of ancient DNA. 20 years ago, what we are doing now would have been impossible, explains Dr. Isaac Barton, a molecular biologist specializing in ancient pathogens. DNA degrades over time. It fragments into tiny pieces. Extracting and sequencing ancient DNA, particularly from pathogens, is like trying to reassemble a million-piece puzzle after most of the pieces have disintegrated. But advances in DNA sequencing technology and bioinformatics have revolutionized the field. Scientists can now extract, amplify, and reconstruct genetic material that has survived for thousands of years. By targeting dental pulp, the soft tissue inside teeth that is often protected from environmental contamination, researchers can recover DNA from ancient bacteria, including Yersinia pestis, the bacterium responsible for plague. Teeth are like time capsules, Dr. Barton continues, holding up a molar carefully extracted from a 14th century skeleton. The enamel protects the interior like a vault, sometimes preserving bacterial DNA from infections that were active when the person died. In 2019, a team led by Dr. Tara Kendrick from the Max Planck Institute for the Science of Human History decided to put the Kyrgyzstan cemetery to the test. They obtained permission to extract dental samples from several of the skeletons buried at the site in 1338 to 1339. Dr. Kendrick recalls the moment vividly. "We were working in a small, specialized ancient DNA laboratory. Everything had to be meticulously cleaned to avoid contamination. Modern DNA, even a single cell from a researcher's skin, could ruin the entire analysis. Wearing full-body protective suits, face masks, and double layers of gloves, the team carefully drilled into the ancient teeth to extract the pulp. The samples were then processed to isolate any preserved DNA, which was subsequently sequenced using high-throughput methods. When the sequencing results came back, we were stunned," Dr. Kendrick says. "Within the dental pulp of these individuals who died in 1338 to 1339, we found unmistakable genetic signatures of Yersinia pestis, the plague bacterium. Not only that, but we could reconstruct its entire genome." The question that had eluded historians for centuries was suddenly answerable through DNA. Was this the same strain of plague that would devastate Europe years later? The answer was about to change history. The laboratory hummed with anticipation as Dr. Kendrick and her team began comparing the genetic sequence extracted from the Kyrgyzstan burials with known plague genomes from later European outbreaks. Using complex phylogenetic analysis, essentially creating a family tree of bacterial strains, they made a startling discovery. The strain we found in these burials was not just related to the European Black Death strain, it was the direct ancestor, explains Dr. Kendrick, pointing to a computer screen displaying a complex branching diagram. Every strain of plague that caused devastation across Europe and beyond could be traced back to this precise variant. We had found the genetic source of the Black Death. This was extraordinary. The evidence suggested that the Black Death pandemic that ravaged Europe from 1347 to 1351 originated from this precise location in Central Asia nearly a decade earlier. But the discovery raised more questions than it answered. How did the disease spread? Why did it become so virulent? And perhaps most importantly, how did a local outbreak transform into the most devastating pandemic of pre-modern times? To understand these questions, we need to dive deeper into the nature of the plague itself. Yersinia pestis is a bacterium that primarily infects rodents and their fleas. Humans are accidental hosts, typically infected when bitten by a flea that has previously fed on an infected animal. The bacterium has been responsible for 3 major plague pandemics in human history: the Justinian plague, 541 to 549 CE, the Black Death, 1346 to 1353, and the modern plague, beginning in the 1860s. What makes Yersinia pestis so dangerous is its remarkable adaptability, explains Dr. Barton. The bacterium can infect a wide range of hosts, from rats to humans, and can be transmitted in several different ways. The most common form of plague is bubonic, characterized by painful swollen lymph nodes called buboes, the black swellings that gave the Black Death its name. Without treatment, bubonic plague kills 50 to 70% of those infected. But the bacterium can also infect the lungs, causing pneumonic plague, which is transmissible directly from person to person through respiratory droplets, and is almost universally fatal without modern antibiotics. The pneumonic form is particularly terrifying, notes Professor Kaplan. It spreads like a cold but kills like Ebola. In medieval times, once the disease shifted to the pneumonic form, mortality rates in affected communities could approach 100%. But the Kyrgyzstan discovery raised an important question. If the Black Death originated in Central Asia in the late 1330s, why did it take nearly a decade to reach Europe? The answer lies in both geography and circumstance. The region around Lake Issyk-Kul was situated on an important artery of the Silk Road, the ancient network of trade routes connecting East Asia with the Middle East and Europe. Merchants, diplomats, and travelers regularly passed through the area, creating perfect conditions for a disease to spread. We believe the plague moved west along trade routes, carried by infected rodents stowed away in caravans or by merchants who themselves became infected, explains archaeologist Dr. Victor Mitchell, who participated in the original excavation of the Kyrgyzstan cemetery. But the disease did not simply travel in a straight line at a constant pace. Its spread was shaped by geography, climate, local rodent populations, and human movement patterns. Ecological factors likely played a crucial role. Plague establishes itself in local rodent populations, creating what scientists call a reservoir. These reservoirs can persist for years or even decades, occasionally causing outbreaks when conditions are right. Think of it as a smoldering fire that occasionally erupts into a blaze. Dr. Barton explains. The bacterium could have traveled westward, establishing itself in multiple rodent reservoirs along the way, before finally reaching a tipping point that allowed for the massive European outbreak. Climate may have been another critical factor. The 14th century marked the beginning of what historians call the Little Ice Age, a period of cooling in the northern hemisphere. Changing climate conditions could have affected rodent populations and their movements, potentially driving plague-carrying rats toward human settlements as their natural habitats became less hospitable. But perhaps the most important factor in the Black Death's devastating spread was the interconnectedness of the medieval world itself. By the 14th century, trade networks spanning Europe, Asia, and North Africa had created unprecedented connectivity between regions previously isolated from one another. The 14th century was, in some ways, the first age of globalization, Professor Kaplan observes. Ships and caravans moved goods, and with them diseases, across vast distances at unprecedented rates. Once the plague reached the Mediterranean, ships became vectors that rapidly spread the disease to port cities throughout Europe. This maritime spread was devastatingly efficient. In October 1347, plague-carrying ships docked in Messina, Sicily. By January 1348, it had reached Marseille in France. By June, it was in Paris. By November, London. The Black Death moved across Europe at an average of 2 miles per day, remarkable speed for the medieval period. The impact was apocalyptic. Contemporary accounts describe cities where the living were too few to bury the dead. In Florence, the chronicler Marchione di Capostefani wrote that all the citizens did little else except to carry dead bodies to be buried. In Siena, Agnolo di Tura recorded how father abandoned child, wife husband, one brother another, and no one could be found to bury the dead for money or friendship. The scale of death is difficult to comprehend. Some cities lost 60 to 70% of their population in a matter of months. Villages were completely abandoned. Fields lay fallow as there was no one left to tend them. The demographic collapse was so severe that it took Europe 200 years to regain its pre-plague population. But while the devastation in Europe is well documented, the impact on the region where the pandemic originated remained largely unknown, until now. The ancient cemetery near Lake Issyk-Kul tells its own tragic story. Archaeological evidence suggests the community was decimated by the plague outbreak of 1338 to 1339. Of the 467 burials identified from this period, more than 100 carried inscriptions explicitly mentioning pestilence as the cause of death. What's remarkable is the concentrated time frame, notes Dr. Mitchell. We see a massive spike in burials within a few months, a clear sign of an epidemic event. And these weren't simple graves. Many contained multiple bodies, suggesting that entire families died together. To better understand what happened in this Central Asian community, researchers turned to the ancient DNA not just of the pathogen, but of the victims themselves. Dr. Kendrick's team extracted human DNA alongside the plague bacterium from the dental samples. This allowed them to reconstruct aspects of the victims' lives, their ancestral origins, potential genetic relationships, and even certain physical characteristics. The community was remarkably diverse, Dr. Kendrick explains. We found genetic markers indicating ancestry from across Asia and the Middle East, confirming that this was indeed a cosmopolitan trading post along the Silk Road. One particularly poignant discovery came from a family burial containing a man, a woman, and 2 children. DNA analysis confirmed they were indeed a biological family. All 4 had died within days of each other, victims of the same outbreak that would eventually transform into the Black Death. When we look at the molecular clock, essentially the rate at which mutations accumulate in the bacterial genome, we can trace how the pathogen evolved as it spread, explains Dr. Barton. The strain found in these individuals from 1338 to 1339 sits at a critical juncture in the Yersinia pestis family tree. It represents what we call the Big Bang, a moment of rapid diversification that gave rise to most of the plague strains that circulated in the following centuries. This Big Bang in plague evolution likely occurred shortly before or during the Kyrgyzstan outbreak. The bacterium had acquired specific genetic changes that may have made it more virulent or more transmissible, the perfect biological storm. But what caused this evolutionary leap? What transformed a relatively localized disease into a pandemic capable of killing half the population of the most densely populated continent on Earth at that time? The answer might lie in the bacterium's genome itself. When scientists compared the Kyrgyzstan strain with earlier and later variants of Yersinia pestis, they identified several key genetic changes. These included mutations affecting the bacterium's ability to infect fleas and to survive in human hosts. There's a specific gene we identified that had undergone a significant change, Dr. Barton explains, pointing to a region on a genetic map. This gene helps the bacterium survive inside human cells. The mutation may have enhanced this ability, making the infection more severe and possibly extending the period during which a person could transmit the disease. Another crucial finding was that the strain had acquired genetic changes that allowed it to more effectively colonize the flea's digestive system, causing blockages that made the flea bite more aggressively and regurgitate more bacteria into the wound, a more efficient transmission mechanism. Evolution gave this strain the perfect combination of traits, Dr. Kendrick observes. It could spread more efficiently between fleas and humans, survive better within the human body, and potentially shift more readily between bubonic and pneumonic forms. It was, in evolutionary terms, a perfect pathogen. But genetics alone cannot explain the catastrophic spread of the Black Death. Historical context provides the other half of the story. The 14th century was a time of significant climate change, population pressure, and political instability across much of Eurasia. The Mongol Empire, which had controlled vast territories from China to Eastern Europe, was fragmenting. This political instability led to conflicts that disrupted traditional ways of life and drove population movements. People were on the move in numbers not seen for centuries, Professor Kaplan explains. Refugees fleeing conflicts, merchants seeking new markets as old trade networks collapsed, armies on campaign, all provided vectors for disease to spread. Additionally, the early 14th century saw a series of famines across Europe and Asia, weakening populations and making them more vulnerable to disease. The Great Famine of 1315 to 1317 killed millions across Europe, creating a population already physically compromised when the plague arrived 3 decades later. We're looking at a perfect storm of biological and social factors, Professor Kaplan continues. A bacterium that evolved to become more virulent and transmissible, moving through populations that were already weakened by malnutrition and social stress, traveling along well-established trade routes connecting previously isolated regions. This convergence of factors explains not only why the Black Death was so devastating but also why it spread in the pattern it did, following trade routes, decimating urban centers before rural areas, and moving at different speeds across various regions. The evidence from Kyrgyzstan provides a crucial missing piece in this complex puzzle. It confirms that the Black Death did indeed originate in Central Asia, but through a specific evolutionary event that occurred around 1338, creating a strain with unprecedented pandemic potential. What we're seeing is the birth of a superbug, Dr. Barton says, a moment when natural selection produced a pathogen with the right genetic tools to exploit the particular social and environmental conditions of the 14th century. But the implications of this discovery extend far beyond historical curiosity. Understanding how the Black Death evolved and spread provides vital insights for managing disease threats today. As Dr. Kendrick walks through her laboratory, she gestures to a wall covered with phylogenetic trees, branching diagrams showing the evolutionary relationships between different strains of Yersinia pestis. The Black Death strain was not the end of the story, she explains. It gave rise to multiple lineages that continued to cause outbreaks across Eurasia for centuries. Some of these lineages persist today in rodent reservoirs around the world. Indeed, plague never truly disappeared. After the initial catastrophic pandemic of 1346 to 1353, the disease returned in waves across Europe and Asia for the next 500 years. The second pandemic, as historians call it, included the Great Plague of London 1665 to 1666, and numerous other outbreaks that periodically devastated communities until it mysteriously receded from Europe in the early 18th century. Then, in the 1890s, a third pandemic emerged from China, spreading to India where it killed millions and eventually reaching every continent except Antarctica. This third pandemic, driven by steamships and railways that connected the world more tightly than ever before, finally subsided in the mid-20th century with the advent of antibiotics and modern public health measures. But plague remains endemic in rodent populations across parts of Asia, Africa, and the Americas. In the United States alone, an average of 7 human cases occur annually, primarily in rural areas of western states. Globally, between 1,000 and 2,000 cases are reported each year. Yersinia pestis is remarkably persistent, Dr. Barton notes. It establishes itself in rodent reservoirs where it can remain for centuries, occasionally spilling over into human populations. This persistence raises an uncomfortable question. Could the Black Death happen again? In terms of the exact same scenario, no, Dr. Barton reassures. We have antibiotics that are effective against Yersinia pestis, and modern surveillance systems can identify outbreaks before they become widespread. But the bacterial genome continues to evolve, and antibiotic resistance is a growing concern. More worrying is what the Black Death teaches us about how pandemics emerge and spread. The conditions that allowed the Black Death to become so devastating have modern parallels, explains Professor Kaplan. Global connectivity, climate change affecting animal habitats and migration patterns, political instability driving refugee movements. These factors can still converge to create pandemic conditions. Indeed, the COVID-19 pandemic demonstrated how rapidly a new pathogen can spread in our interconnected world. While the mortality rate was far lower than the Black Death, the fundamental dynamics of global spread followed similar patterns, moving along transportation networks, exploiting human connectivity, and affecting different populations in different ways based on social and environmental factors. What the Kyrgyzstan discovery teaches us is that pandemics often begin in unexpected places, under specific conditions that allow a locally circulating pathogen to acquire pandemic potential, Dr. Kendrick observes. The challenge is identifying these potential hotspots before a pathogen makes the evolutionary leap to become a global threat. This is where the study of ancient pandemics offers crucial insights. By understanding how the Black Death evolved and spread, scientists can better predict how modern pathogens might behave under similar pressures. The technology that allowed researchers to extract and analyze 700-year-old bacterial DNA is now being applied to monitor contemporary disease threats. Genomic surveillance, tracking the genetic changes in circulating pathogens, has become an essential tool in public health, allowing scientists to identify concerning mutations before they lead to more transmissible or virulent strains. During COVID-19, we sequenced viral genomes in almost real time, tracking new variants as they emerged, Dr. Barton explains. This is the same approach we used to reconstruct the evolution of the Black Death, but applied proactively rather than retrospectively. But beyond the scientific implications, the discovery in Kyrgyzstan offers something more profound, a human connection across 7 centuries. In a climate-controlled storage facility at the University of Tübingen, Dr. Mitchell carefully removes a small stone from a protective container. It is a headstone from the Kyrgyzstan cemetery, inscribed in a form of Old Turkic script. This commemorates a man named Sanmak who died in the year of the tiger when pestilence was in the community. Dr. Mitchell translates. It was commissioned by his brother, who was clearly literate and wealthy enough to afford a carved stone monument. For Dr. Mitchell, this personal connection is what makes the discovery so powerful. These weren't just anonymous victims of a historical event. They were individuals with names, families, and stories. Sanmak wasn't thinking about being patient zero in what would become the worst pandemic in history. He was a person whose life was cut short by a disease no one understood. The genetic analysis of the victims reveals other intimate details. Some showed evidence of prior health challenges, healed fractures, signs of nutritional stress during childhood, indicators of previous infections. One young woman had given birth shortly before dying. A child buried with elaborate grave goods suggested a family of high social standing. Through DNA and archaeology, we can begin to reconstruct not just how these people died, but how they lived. Dr. Kendrick explains. We can see evidence of their diets, their ancestry, their health challenges throughout life. They become real to us across the centuries. This human connection extends beyond the victims to their communities. Archaeological evidence from the cemetery and surrounding area suggests that the local outbreak was devastating but not apocalyptic. Life continued in the region after 1339. Trade resumed. New people moved in to replace those who had died. Communities are remarkably resilient, Professor Kaplan observes. We see this pattern repeated throughout history. Even after catastrophic population loss, societies rebuild. New social structures emerge, often quite different from what came before. Indeed, the Black Death triggered profound social changes across Europe. Labor shortages following the pandemic led to higher wages for survivors and accelerated the end of serfdom in Western Europe. The psychological impact of mass death contributed to changes in religious practices and artistic expressions. Some historians even link the plague to the early stirrings of the Renaissance, as the shock of massive mortality forced a reevaluation of medieval worldviews. Major pandemics aren't just biological events. They're inflection points in human history, Professor Kaplan explains. They reshape societies in ways that can take centuries to fully manifest. The Kyrgyzstan discovery adds an important dimension to this understanding by showing how a localized outbreak in Central Asia transformed into a global catastrophe through a combination of biological evolution and human connectivity. It's a reminder that pandemics don't respect borders or cultural boundaries. Dr. Kendrick notes, the people buried in this cemetery couldn't have imagined that the disease affecting their community would soon devastate populations thousands of miles away. They were the first links in a chain of transmission that would ultimately encompass much of the known world. This interconnectedness across time and space gives the discovery particular resonance today. As we face our own pandemic challenges and the ongoing threat of emerging diseases, the lessons from 14th century Kyrgyzstan seem increasingly relevant. When we extract ancient DNA, we're not just studying history, Dr. Barton reflects. We're learning how pathogens evolve, how they spread, and how they interact with human societies. These lessons are directly applicable to managing disease threats today and preparing for those we'll face tomorrow. The discovery also reshapes our understanding of global history. For centuries, the narrative of the Black Death has been Eurocentric, focusing primarily on its impact in Western Europe. The identification of its Central Asian origins helps restore a more balanced perspective, highlighting the interconnected nature of the medieval world and the central role of regions often marginalized in traditional historical accounts. This isn't just about correcting the historical record, Professor Kaplan emphasizes. It's about recognizing that global health challenges have always required global perspectives. The Black Death wasn't a European disease that came from somewhere else. It was a Eurasian pandemic that affected diverse populations across multiple continents. As researchers continue to analyze the genetic material from Kyrgyzstan, they hope to answer even more questions about the birth of the Black Death. Was there something unique about this community that facilitated the evolution of a more virulent strain? Were there environmental factors specific to this time and place that contributed to the bacterium's transformation? And perhaps most intriguingly, were there other outbreaks in Central Asia that failed to spark global pandemics, and if so, why? We're just scratching the surface, Dr. Kendrick admits. There are likely other burial sites across Central Asia with similar stories to tell. Each new discovery helps us build a more complete picture of how this catastrophic pandemic emerged and evolved. As the sun sets over the shores of Lake Issyk-Kul, the medieval cemetery lies quiet once more. Many of its secrets have been revealed, but many remain buried. The stones marking the graves of those who died in 1338 to 1339 stand as silent witnesses to a pivotal moment in human history, the birth of a pandemic that would reshape the world. In her Cambridge laboratory, Dr. Blackwell holds up a vial containing extracted DNA from one of these ancient victims. This is where science becomes almost philosophical, she muses. In this tube is genetic material from both a human who died nearly 700 years ago and the bacterium that killed them. Through this molecular connection, we can reach across centuries to understand not just how they died, but how their death connects to millions of others across time and space. This connection between past and present takes on added significance as we navigate our own pandemic challenges. The discovery of the Black Death's origins reminds us that we are part of an unbroken chain of human experience stretching back through centuries of triumphs and tragedies, innovations and catastrophes. What strikes me most about this research, Professor Kaplan reflects, is how it collapses time. Suddenly, the 14th century doesn't seem so distant. We can see ourselves in these people, their vulnerability to disease, their struggle to understand what was happening to them, their resilience in the face of catastrophe. Indeed, the parallels between the Black Death and modern pandemic experiences are sometimes uncanny. Medieval communities implemented versions of quarantine, debated the causes and cures of the disease, and experienced both social fragmentation and remarkable solidarity in the face of mass mortality. Reading accounts from the time, you see the same human responses we witnessed during COVID-19. Professor Kaplan continues. Fear leading some to isolate completely, others denying the severity of the threat, communities coming together to care for the sick, and authorities struggling to balance public health with economic and social needs. These human continuities across 7 centuries provide both perspective and humility. For all our technological advances and scientific understanding, we remain vulnerable to the emergence of new pathogens or the evolution of existing ones. The Black Death arose from a specific confluence of biological and social factors, Dr. Barton explains. Different pathogens exploit different vulnerabilities in our interconnected world. Understanding these patterns helps us prepare for future threats, but we can never eliminate risk entirely. This sobering reality is balanced by the remarkable scientific progress that allowed the discovery in the first place. The ability to extract, sequence, and analyze 700-year-old bacterial DNA represents a technological achievement that would have seemed like science fiction just a few decades ago. When I began my career, we could barely sequence modern bacterial genomes, Dr. Barton recalls. Now we're reconstructing the genetic code of pathogens that existed centuries ago, tracking their evolution across time with precision that was previously unimaginable. This scientific capability gives us advantages our medieval predecessors lacked. We can identify pathogens quickly, understand their biology, develop targeted treatments and vaccines, and implement evidence-based public health measures. The Black Death spread for years before anyone understood what was causing it, Dr. Kendrick notes. Today, we can identify a new pathogen within days, or even hours of an outbreak beginning. That's an extraordinary advantage. Yet technology alone cannot protect us. The social, economic, and political contexts in which diseases emerge and spread remain crucial factors in determining their impact. What the Kyrgyzstan discovery teaches us is that pandemics are never purely biological events, Professor Kaplan emphasizes. They arise from complex interactions between pathogens, environments, and human societies. Addressing only the biological dimension will never be sufficient. This holistic understanding, bridging natural science, social science, and humanities, represents perhaps the most important lesson from the discovery of the Black Death's origins. Effective pandemic prevention and response require integrating knowledge across disciplines and scales, from the molecular to the global. As research continues, scientists hope to identify additional archaeological sites that might contain evidence of the Black Death's emergence and early spread. Each new discovery adds pieces to the puzzle, helping to construct a more complete picture of how the most devastating pandemic in recorded history unfolded. We're rewriting history with molecular evidence, Dr. Mitchell reflects. Each ancient genome we recover provides data points that no chronicle or historical account could offer. We're establishing ground truth about what actually happened, rather than what people at the time believed or recorded. This ground truth has profound implications not just for understanding the past but for navigating the future. By establishing precisely how, when, and where the Black Death emerged, scientists can better identify the conditions that might give rise to similarly devastating outbreaks in the modern era. In a high-security laboratory at the Centers for Disease Control and Prevention in Atlanta, researchers monitor genetic changes in pathogens collected from around the world. The lessons from the Black Death directly inform their work, helping them recognize patterns of evolution that might signal increased pandemic potential. Ancient DNA research gives us baseline knowledge about how pathogens evolve under natural conditions, explains Dr. Barton, who consults with the CDC on emerging disease threats. When we see similar patterns in modern surveillance data, it raises red flags. This integration of historical research with contemporary public health represents a powerful new approach to pandemic preparedness. Rather than treating historical outbreaks as interesting but irrelevant curiosities, scientists now recognize them as case studies providing vital insights into disease dynamics that remain relevant today. The bacteria and viruses that caused historical pandemics evolved according to the same biological principles that govern modern pathogens, Dr. Kendrick explains. Understanding these evolutionary patterns helps us predict how current threats might transform in the future. For the communities near Lake Issyk-Kul, the research has brought renewed attention to their historical significance. Local museums have developed exhibitions explaining the region's connection to the Black Death, and archaeological tourism has increased as visitors seek to understand this pivotal moment in global history. There's a certain pride in knowing your region played such an important role in history, even if it's a tragic one, notes local historian Murat Aliyev. It connects our community to the broader human story in a way that transcends borders and centuries. This connection across time and space is perhaps the most profound outcome of the discovery. Through ancient DNA, we have established a direct link between a specific community in 14th century Central Asia and millions of victims across multiple continents in the years that followed. It's humbling to think about, Dr. Blackwell reflects. The bacterium that infected these individuals in 1338 was the ancestor of the strain that would kill millions throughout Eurasia and North Africa. Through genetic analysis, we can trace that lineage with remarkable precision. As the research team publishes their findings in scientific journals and presents at conferences, the story of the Black Death's origins continues to evolve. New analyses of the genetic data reveal additional details about how the bacterium transformed from a relatively localized threat into a global catastrophe. We're now looking at specific genetic changes that occurred as the bacterium spread westward, Dr. Kendrick explains. We can see it adapting to new environments, new host populations, perhaps even becoming more virulent as it moved into Europe. This evolutionary tracking provides a window into how pathogens respond to new opportunities for transmission and new selective pressures. It reveals the bacterium not as a static entity but as a dynamic organism adapting to changing circumstances, a perspective that helps explain why diseases emerge, spread, and sometimes disappear. Pathogens and hosts are engaged in an evolutionary arms race, Dr. Barton explains. As humans develop immunity, either naturally or through medical interventions, pathogens evolve to overcome those defenses. Understanding this dynamic is crucial for managing disease threats over time. For the victims buried near Lake Issyk-Kul, this scientific understanding would have seemed miraculous, an ability to see the invisible forces that were devastating their community. For modern scientists, it represents both a remarkable achievement and a sobering responsibility. We now have the tools to understand pandemics in ways our ancestors could never have imagined, Dr. Blackwell observes. With that knowledge comes an obligation to use it to prevent similar catastrophes in the future. As the sun rises over Cambridge University, Dr. Blackwell returns to her laboratory to continue her work. On her desk sits the ancient tooth, a silent witness to a pivotal moment in human history, now yielding its secrets through the power of modern science. In the end, this isn't just about bacteria or genomes or archaeological sites, she reflects. It's about understanding our shared vulnerability and resilience as a species. The Black Death was the most devastating pandemic humans have ever faced, yet we survived. We rebuilt. We carried on. This perspective offers both caution and hope as we face our own pandemic challenges. The discovery of the Black Death's origins reminds us that we remain vulnerable to the emergence of new pathogens, but also that we have unprecedented tools to understand and combat them. History doesn't repeat, but it rhymes, Professor Kaplan observes, quoting Mark Twain. Each pandemic has its own unique characteristics, but certain patterns recur. By understanding these patterns, we can better prepare for what might come next. As the ancient DNA from Kyrgyzstan continues to reveal its secrets, it bridges 7 centuries of human experience, connecting us to ancestors who faced their own version of the challenges we confront today. Through science, their story becomes part of our story, a testament to both human vulnerability and human resilience in the face of invisible threats. In the teeth of the dead, we find not just the origins of the Black Death but reflections of ourselves, our fears, our courage, our capacity to endure and rebuild in the aftermath of catastrophe. This connection across time offers perhaps the most valuable lesson of all, that even in our darkest hours, we remain part of an unbroken human story that stretches both behind and before us, a story of struggle and survival that transcends the boundaries of time and space. Ancient DNA does not just expose the mystery of the Black Death's origin. It illuminates our shared humanity across the centuries, teaching us that in our vulnerability to disease and our determination to overcome it, we are one with generations past and generations yet to come. In the microscopic fragments of bacterial DNA preserved in the teeth of those who died in a remote Central Asian community in 1338, we find not just scientific facts but profound truth, that the invisible threads connecting human lives across time and space are as real and enduring as the genetic code itself.